would just remind you to remain muted during the presentation, and then we'll address questions at the end. So I'd like to introduce Ken Burns, who will introduce our speaker for today. Thank you for joining.
Thank you, Julie. So we're delighted to have Piyush Jain here. He's a little different than most of the speakers in this course because he's not a grizzled old so-and-so, but in fact, he's an assistant professor in chemical engineering who's been at the University of Florida since 2018. He did his undergraduate work at the Central University in India, and then he moved to the University of Missouri in Kansas City to receive his PhD in pharmaceutical sciences with a minor in chemistry. And he must have done pretty well because he went straight from there to a postdoctoral position at MIT with Professor Bhatia, and where he worked on CRISPR nanotechnology. So it was very good preparation, at least for today's lecture, and presumably some other work that he's doing as well. He's been here at UF since 2018. As I said, he's an assistant professor in chemical engineering, but he also, since 2021, has had an endowed chair called the Shah Star Endowed Chair for Promising Young Faculty, named after Dinesh Shah, who is a longtime professor of engineering here at the university and very successful. So in his job, he is also, Dr. Jain is also the principal investigator of the Laboratory of Nanobiomedical Precision. And they're developing biomolecular delivery systems, applying gene editing technologies for diagnosis of infectious and genetic diseases and cancer. And currently, he's gotten very applied and he's developing home testing kits for COVID virus, which is an interesting approach and has achieved some recognition. Anyway, this year's last year's 2020 Nobel Prize was awarded for work on CRISPR to Professor Carpentier, who's in Germany, although she's from France, and to Jennifer Doudna, who's at the University of California, although she's from Hawaii originally. And she's been on the East Coast quite a bit as well before she got out to Berkeley. So this is, I would say, to me, this prize in 2020 was what I call a no-brainer. I was just waiting for somebody to get the prize, and Jennifer and Michel Charpentier were clearly two of the really big early names in the field. I think that it was very appropriate that they got it. Now, I just want to say one thing before he starts. There was, of course, a rush to patent the use of CRISPR for whatever. And there were a couple of guys from Harvard who got into this early and applied for a patent. And there was a fairly long legal struggle. I don't even know if it's been resolved yet. Maybe Piyush can tell us. But one of the Harvard people who's a certified genius named George Church was asked whether he felt it was appropriate that Jennifer and Michelle got the prize. And he said, you know, it's absolutely appropriate because the prize is for discovery, not for application. And what George Church and his compatriots were working on was application. So I thought that was a very gracious response to a kind of a tough question from some reporter. Anyway, enough. Piyush, it's up to you. Please take over and welcome.
Thank you so much, Ken. It's been a pleasure, and thanks for a great background about me, and I hope I can do the justice. This field of CRISPR is very dear to me, and this Nobel Prize. Oh, this must be an auto mode. Let me fix that first. So this field is definitely. Let's see. Just okay. Yeah, so my lab actually focuses on CRISPR-Cas technologies and development of these technologies. So this field is very dear to me, and we call our lab as editing the editors. And when we say we are not editing the journal editors, right, or newspaper editors, we are talking about gene editors. So we are the ones who actually edit the gene editors so that we can improve them better. And so this is just my lab. I'm here in the CGRC building, and I'm really delighted to talk about CRISPR technology. So as Ken mentioned, this year's Nobel Prize was given to two brilliant scientists, women scientists who won this award and shared equally for the development of a method for gene editing called CRISPR, right? And so I will tell you about my story, how I learned about CRISPR, and then we can talk about what it is and where it's going, the field is going. And also I'll discuss about the patent issues going on with CRISPR, as well as some of the things that we are already worried about, like ethical concerns related to gene editing in babies and embryos. As Dr. Burns already mentioned, so I did my undergrad from India, moved to the US in 2006, and since 2018 I've been here as a faculty. In between I was at MIT, and I have a story about, you can see when I will tell you about like how I learned about CRISPR. Is basically after I did my PhD, I went on to interview for postdoc positions, and that's when I learned about CRISPR. And so my current lab focuses on three different areas. We are improving the CRISPR technology. We are trying to deliver it better so that we can apply it. And then also we are doing diagnostic work as well as therapeutics work. So we are all working with CRISPR technologies and trying to improve and apply. So this is my story in 2013, January 8th, 2013. I had a call from David Liu, who is now a CRISPR expert. He is at Harvard and also HHMI member. He called me for a postdoc interview, and I was really excited about the interview. And then after his call, I had another call for an interview with Professor Sangeeta Bhatia, whose lab I joined eventually. And so this interview was on January 10th, so 2 days apart. Okay, so 8th and 10th. And January 9th was my birthday. So I was actually waiting in the hotel room, and I got an email from this guy, Jack Sostek. And so he emailed me, he's a Nobel laureate and a 2009 Nobel Prize winner, and also doctoral advisor of Jennifer Doudna. So he emailed me, and then he told me like, well, when can you come visit Boston? And he's also located at Harvard. And I was like, okay, I'm in Boston. And he's like, why don't you come down? And so we had a phone conversation right away. And then, I just dressed up and got to his office. And then I met with him, and we spoke for 2 hours. And during our conversation, he brought up CRISPR to me. And it just happened that this interview was on in 2013, and I had heard about CRISPR, but I had no knowledge because I had not read about CRISPR, what it is exactly, how it works. I had no idea. And so he was super excited by Jennifer Doudna's paper, and he mentioned, did you see that paper in Science? I was like, no, I have not. And so he explained to me how it works. So I got to learn about CRISPR from a Nobel laureate, from Jennifer Doudna's advisor. So that was, that was really cool. And so I learned more and more, but I had two offers. And so I decided to join Sangeeta's lab instead, because um, I felt like she was doing translational work, and my background has been in pharmaceutical sciences, as Ken mentioned. So I felt like I would be better fitted there. So Sangeeta, since then, also rose, and she is now in 4 national academies, like National Academy of Sciences, Medicine, Engineering, and inventors. So she's doing great. And so I joined her lab, and then also I want to mention that this is the quote from Jack Sostek's work from his Nobel lecture, when he talked about Jennifer Doudna in his Nobel lecture, and in his notes. So what he said, that Jennifer was the first student who worked on this problem of ribozyme, and these are enzymes which are made up of RNA, and they were discovered by this Thomas Cech's group. Okay, and so he convinced Jennifer to work on this RNA biology. And even though she was really interested in working on yeast genetics, and so eventually Jennifer Doudna joined Thomas Cech's group as a postdoc after she graduated as well, so at UC Boulder. So there's a connection of all the people. And then from Sangeeta's group, I met with Fang Zheng's group across the street at Broad Institute. And then I started collaborating with his group also. And we have worked also, and we are right now also working together as well. So I've been very fortunate to have met big shots in the field. And so what happened is that the day Nobel Prize was announced, I was sleeping, and I just got a call in the morning from a reporter saying that, did you hear the news? I was like, what? Nobel Prize was awarded to CRISPR, and I want you to write a piece. So I just wrote a piece on this conversation about CRISPR, and I was really excited. In fact, every year I've been thinking that CRISPR will win the prize this year, and it's just that it kept delaying. But it was very interesting that it was, of course, it was no brainer that it will be awarded, but it was very difficult question to whom to award. And of course, Jennifer Doudna and Emmanuel Charpentier were like the top people. So this is just the history of CRISPR. This is the timeline. So CRISPR started in 1987, where Japanese scientists discovered these sequences in the genome. And so the CRISPR is actually a genetic sequence, which we'll come and talk about in a few minutes. And then it was found that these CRISPR sequences exist in widespread in a lot of different prokaryotes. And then the name was given to these as CRISPR, and I'll talk about what CRISPR stands for. And then most of this work was done on figuring out what CRISPR does. Why is that exist? And so they figured out that it might be responsible for adaptive immunity. So in about 2007, they injected CRISPR sequences in bacteria, and they found that it can adapt immunity. And so since then, people have been interested in CRISPR, but there are not a lot of papers and publications. And people also discovered, so this was a seminal paper in 2012 when Jennifer Doudna's group published this paper. And since then, there are about 23,000 publications or so on CRISPR. So this field especially took off in 2012, after 2012, when this was published from Jennifer Doudna's group. In 2013, it was used for eukaryotes. And the first clinical trial for CRISPR was done in 2016. And there are many clinical trials, actually 46 going on right now in the US. And so CRISPR-Cas systems what it is. So it's a complicated system to understand, but now that we understand much better than before, I can explain you. So just like us, bacteria can get flu. So when they get flu, the virus attacks the bacteria, and they would like to, the virus would like to multiply inside the bacteria and use its resources, and then kill the bacteria eventually, right? And so bacteria doesn't like it, and so they have evolved a system to defend themselves. In order to defend themselves, they have this system called CRISPR. That system allows them to fight against the virus. What it does is, when a virus invades the bacteria takes a tiny piece of its DNA and stores that information into its own genome, so that they know that this virus came before and it had invaded before. And so this information is stored every time any virus or foreign DNA invades the bacteria. Keeps this information in a tiny array, okay? And this array is known as CRISPR array. So these, when they sequenced this for the first time, they found that these sequences are oftentimes, these are palindromic in nature. So there are sequences that you see in diamond. These are repeated sequence. So they are exact same sequence that are repeated, but they are oftentimes flanked by these non-repeat sequence. So those colored rectangles indicate the sequences that are from different viruses and foreign DNA that invaded bacteria. And so these sequences are all aligned in this CRISPR array. And then scientists also discovered that these arrays don't make any proteins. It's just there, but they don't have the capability of making the protein. However, they can make RNA out of this. So when this array gets transcribed, they make RNA. And in addition to this, there is a there are upstream proteins, and sometimes also downstream. There are proteins that are associated with CRISPR. So these are known as Cas proteins. So CRISPR stands for clustered regularly interspaced short palindromic repeats. So it's a cluster of regularly interspaced. So these are called spacers, and they are of the same length, and they are regularly interspaced. And they are talking about these diamond shaped structure. These are the repeat sequences, and they are also palindromic. Okay, so these are the clustered regularly interspaced short palindromic repeats, and these repeat sequences are known as CRISPR. And they had no idea why they exist in this bacteria. And then when they looked at other organisms like archaea, they also discovered that now CRISPR systems have also been discovered in large bacteriophages. So these sequences exist in our animal kingdom, or in the prokaryotic system especially. So what happens with this system is that the Cas locus, which makes these proteins, they give rise to this Cas protein, and the CRISPR array give rise to the pre-CRISPR RNA. And the pre-CRISPR RNA are these long RNAs, and because they are palindromic, they make these hairpin loop structures. And these hairpin loop structures are now separated by these spacers. And these spacers are from different viruses, remember? And so these pre-CRISPR RNAs are then processed into CRISPR RNAs, and these CRISPR RNAs and Cas proteins, they make complex, and these complex can now bind to the viral DNA. And Cas is a nuclease, so it can cut the viral DNA and causes double-stranded breaks. So in a nutshell, this CRISPR RNA, because they were derived from the viral sequences, they act like a GPS, and they tell Cas protein, which is a nucleus, to where to go. And so this nucleus acts like a scissor. So it's a combination of a GPS and a scissor, which can then tell the bacteria to where to cut. If a virus invades again, this bacteria has already this information, and they can now make double-stranded cuts. Okay, any questions on this?
Can I'd like to ask a question if
Sure.
possible. Um, those 1 to 10 represent 10 different virus, for instance.
Exactly.
So each one of those represents some information about that virus, and it's attached by attached to this palindromic cutting mechanism.
Yes.
And basically, they use that. First, they use the proto that the colored portion to identify
Mhm.
a virus, and then they use the palindromic method to tell the protein that's where you should cut it, and
Exactly.
that kills it. So that sums it up, right?
Yes,
In a
yes.
way.
And there are,
Okay.
yeah, there are nuances. So these are just nomenclature. So before it is integrated into the bacterial genome, it is called
Right.
protospacer. Once it's integrated, they are known as spacers. And
Ah,
then the red sequence you notice here is called
mhm,
protospacer adjacent motif, or PAM. And it's
mhm.
also very important for CRISPR because this PAM only exists in the virus, and when it copies from the virus, this information is not added to the CRISPR array. And that's how bacteria can differentiate between its own genome versus the viral genome. So,
Right.
so if they see this red color thing when this virus reinvades, they know that this is a foreign DNA and we should cut it. And because
Right.
it's not integrated into the CRISPR array, they will not cut its own DNA.
Right.
Yeah.
Very good.
So really cool system,
Yeah.
clever
Yeah.
system, they have evolved.
Uh-huh.
So where does the Jennifer Doudna and Emmanuel Charpentier come? So Emmanuel Charpentier, she figured out a third piece. So I mentioned two pieces here. Okay, so I mentioned the Cas protein and the CRISPR RNA. These two combined together will cut the virus. But there's a third piece which was missing. And the missing piece is that how these pre-CRISPR RNAs get processed into CRISPR RNA. Okay, and what she did was she published a paper in Nature showing that there's a trans-activating CRISPR RNA, which means there's a special RNA called tracer RNA or trans-activating CRISPR RNA. So if you read the nomenclature, CRISPR RNA stands for CRRNA, and then the tracer RNA is basically trans-activating CRISPR RNA. And so if you have a pre-CRISPR RNA, this pre-CRISPR RNA is processed into a specific shorter CRISPR RNAs by using this green trans-activating CRISPR RNA. This trans-activating CRISPR RNA binds, and then the the RNase 3, which is another nuclease, will make cuts and process them into shorter CRISPR RNAs. And interestingly, what was discovered was that this trans-activating CRISPR RNA, when it binds to this pre-CRISPR RNA, it doesn't let it go. And this Cas9 and the CRISPR RNA and the tracer RNA, all three makes a complex together. So so this complex is the active complex. So you need three components. You need a CRISPR RNA, you need a Cas protein, and then you need a helper, which is a trans-activating CRISPR RNA. And so this was what was published by Emmanuel Charpentier. She figured out that RNase 3 is required and tracer RNA structure, she figured that out. And so she published a paper in Nature, and then she collaborated with Jennifer Doudna to do further work to see whether we can cut any DNA and program it to cut any DNA outside the cells. And so what they did also was they connected them together by a linker. So now you have a single piece of RNA, which is known as single chimeric guide RNA, because you have the CRISPR RNA and tracer RNA connected by a linker. This is called as sgRNA. So there are a lot of nomenclatures, but normally when people are now using, these are all, some people call them as guide RNAs because they guide where to go. And so they call it guide RNA. Others will call it as sgRNAs. And so when you see sgRNA, it refers to this single chimeric guide RNA connecting the CRISPR RNA with the tracer RNA. And we have a Cas protein, which acts like a molecular scissor. So this was the paper. So they collaborated together and they won the Nobel Prize, of course. And so if you look at this whole region, you notice that there was a blue region in this CRISPR RNA. So this blue region is the spacer. Okay, this is the region that you can change and program it to cut any DNA. So you just need to change this sequence. The gray region, you can imagine it as a constant region or conserved region. You don't have to change, and you can use that region and keep it same. So only thing you have to change if you want to cut any DNA is just this blue region of the RNA. Okay. All right. So this was the paper that got published in Science, and now they have, it's about 11,000 citations already for this paper. So you can imagine the field has grown significantly because, and this was exponentially, not just significantly, exponential because of this paper, because this is a landmark paper, just came published online in June 2012, and in August 2012 issue in Science. And so after that paper, there are many papers that came out from different labs that showed that you can use CRISPR to do the gene editing in mammalian cells. That was the next paper from George Church group as well as Feng Zheng's group in 2013 January. Came out in the same issue in Science back to back. And so they showed that you can do in mammalian cells. And but this is just showing you now the total number of publications that rose after that. So 2012 is right here. So this is 2012, and then this is going up exponentially. Every year you see a increase. Now we are already in the March of 2021. So I'm sure like this will rise up to, you know, more than 6,000 or 7,000 for sure. So this is great. Every year it keeps rising, and there are 23,000 publications already with using this word CRISPR. So it's hard to capture everything in this talk, but I'll try to summarize, you know, where the field is moving and how things work. So as I mentioned, that you can program CRISPR-Cas system to target any gene in the body. So how it works is that, let's say you know there is a target DNA. This is the gene you want to edit. All you need to know is find out the sequence of the gene and then design your CRISPR RNA and or the guide RNA. And this single chimeric guide RNA, you just change this position. So from blue to green to, you know, whatever you gene you're targeting, you make it complementary to one of the strands of the gene, and look for a PAM sequence. I have not mentioned here, but there's a requirement that there's a PAM sequence as well. So once it finds these two things, as you know, the CRISPR RNA and Cas, they make a complex together. And so they will complex together and then search across the genome. So we have 3 billion base pair. It will search across the entire genome and find where the match is, and then make double-stranded cuts in the DNA, okay? And once the cut is made, cells will immediately come and repair. And most prominent method of repair in our body is called NHEJ, or non-homologous end joining. So they will join the ends of the DNA, and oftentimes cell will grab any nucleotide that is present in the vicinity. So they will oftentimes insert a nucleotide, or they can also delete nucleotides. And this insertion or deletions are known as indels, okay? And so when you make a cut, you don't get back the same DNA. And if you get back the same DNA, it will cut again. So it's like a process until you get a mutated DNA here. And when there's mutations, now the cells have been edited. And so this type of editing you will see is not the one you would like to see for treatment of diseases, but most likely if you imagine applications where you want to disrupt a protein of interest, you can use a CRISPR technology, make a double-stranded cut. Cells will no longer be able to make that protein, because you inserting these nucleotides or deleting these nucleotides will change the protein sequence itself. Sometimes you get this premature stop codon, which means that your protein will not be a full length. You get truncated proteins. Other times you will get modification in the protein. And so what people have thought is that if you can, there is another way, which is if you want to change any DNA to your desired sequence is very hard because cells don't like that. So the way you do it is you just take the DNA sequence of your interest, inject it with the CRISPR and guide RNA inside cells, and then cells will be fooled into copying that information from that DNA template when they are dividing. So when the cells divide, they copy information from, if there is any damage to the DNA, they will copy information from the other pair of the DNA allele or other chromosome, because you have two pairs of chromosome. And if you have a floating template, then they will copy from that. And so you can incorporate your desired modified sequence into the genome. Okay, so why is it important? Because you have so many applications in the field of biotech, including fuel industry, food industry, materials. You can also apply it for medicine. So you can treat diseases, especially genetic disorders. You can use CRISPR-Cas technology for drug development, just understanding how drug functions, and you can modify cells. And also biology, just understanding how different genetic sequences work, or controlling the sequences inside the biology to change the biology. So this has a wide range of applications, and we'll talk about some of these applications today. So again, want to bring back the patent dispute here. So the patent was issued in April 2014 to Feng Zhang's group at MIT, even though Jennifer Doudna's group published first in 2012, June. So this is the timeline what happened. What happened was in June of 2012, they published. They must have filed in May their provisional patent. This was the time before US PTO had a different rule. They had a rule of first to invent instead of first to file. Barack Obama signed an America Invents Act in 2011, and that changed the patent from first to invent to first to file. So now whoever files first wins the patent, and there's no, hopefully there will be less dispute. But this was before that was issued. So it was you can see that in March 2013 is the rule. This rule of first to file was applied in March 2013. So what happened is that they were the first to invent happened before that rule started, right? So you can imagine that's why there is a dispute, because Jennifer Doudna, they published a paper in Science. Feng Zhang's group filed a patent in December 2012. Okay, and they prioritized it. So they filed a provisional patent, but they prioritized the application. So this was provisional, and then they published a paper in how to use CRISPR for editing of mammalian cells. So cells that that are not prokaryotes, eukaryotes, and they found that they published this paper and they had filed already patent, and then in May 2012. It's a wrong listed here, but in May 2012 they have filed. Jennifer Doudna came back and filed the full patent. Okay, so you can imagine in May 2011 in May 2012 they had filed the priority. They had filed the provisional patent, and in March 2013 they filed the full patent. Okay, that makes sense. So in May 2012 they had already filed this patent. In March they filed the full patent, and they used this priority date. Now the rule changed. Okay, and then Feng Zhang's group filed a patent on this applying CRISPR for mammalian gene editing. And this is a huge, billions of dollars are at stake. Imagine, and they also asked the USPTO for accelerated examination. So they paid extra priority fees for the accelerated examination, and the USPTO granted the patent to Feng Zhang's group. And so in 2014, they received their patent, while Jennifer Doudna's patent was still pending. Okay, and then the Jennifer Doudna received the patent in 2014, later 2014-15, and then there was an interference filed in 2016 from UC Berkeley, claiming that the Broad Institute, which was awarded patent through Feng Zheng, is invalid. Okay, they claim that this patent should belong to the UC Berkeley. All right, any questions? It's complicated.
Why should scientific discoveries be open to patent? Aren't they universally accepted as discoveries? Do we patent? Could Galileo have patented the moons of Jupiter? You know,
Yeah.
I'm just wondering. It
No,
seems
you
like
are absolutely right. So this patent they filed was not for scientific discovery, but for invention, showing that CRISPR can be used for editing, DNA editing. And so it can be targeted and you can edit things. Feng Zheng's group showed that you can edit the mammalian cells. And so that was an invention again, but he had to modify certain things in the CRISPR. For example, bacteria doesn't have nucleus, and these systems are evolved in nucleus. Okay, so Feng Zheng's group, when they used CRISPR in mammalian cells, it did not work because our DNA is stored inside the nucleus. So he had to put the nuclear localization signal, which is like a GPS for the nucleus, to the CRISPR-Cas system. Now, they did that and they were successful and they got the patent. Jennifer Doudna claims that this nuclear localization signal is a standard thing. It's not something that he developed in his lab. So it means that anyone could have come and done it. And so there's a fight between the two groups. But you know, there are billions of dollars at stake here. I will come to that and talk about companies, how many companies have built so far. It's amazing around this technology.
okay.
Okay,
Yeah, I think the question is, especially research that's funded by the government, why should any private person be able to have ownership of the results of that research? And the reason is the Bayh-Dole Act. The Bayh-Dole Act specifically stated that if work was achieved or results were achieved from federal funding and it could be applied and the inventor or investigator applied to use it, that they could benefit from it. And the reason was that there was a lot of complaints that all this money was going in to research, exciting things were being found, and nothing happened as a consequence because there were no practical applications that were developed. I can tell you that as soon as money became involved, that situation changed abruptly.
Thank you.
Bob.
I had no idea about this, but yeah, it makes sense.
Bob Rothstein has
Yeah.
a question.
Yeah, I have a question that you may have covered, but I didn't get. I understand how the signal comes in, cut here, attach and cut here, got that. But if you want to, in a eukaryote, if you want to then, after you cut and remove a problematic, the gene sequence, where is the new insertable DNA for the eukaryotes carried? Is it attached to the same complex, or does it have to come in a different complex?
Okay, it's a great question. So the question is that how would you deliver the template, the repair template, right? So for example, after you made the cut and you want to, so there's a cut copy paste function, right? Just like we have it. So we have the cut paste function here. So how would the paste function function? So here, what normally people do is they package everything into a nanoparticle that can carry a repair template, as well as the Cas, as well as the guide RNA. So that's one of the ways that you can deliver your repair template, and the cells will, when they are dividing, they have to be dividing. If they are dividing, then they will copy that information. So several other technologies have been developed where you don't need the cells to divide, and you can still fool the cells to copy paste. So those technologies are still emerging, I would say, but the idea is that you, in your repair template, you also add the CRISPR cutting sequences, and then the cells will cut those also, and then they somehow get confused and they they insert your sequence also. And so that's another way. And then in terms of delivery, you can also deliver these repair templates along in the viral sequences as well. So people have used the AVs also as a source for the donor template. So you can have a donor template which is double stranded, and then have these cut sequences on both the sides, and the CRISPR will be formed, and the AVs, for example, will carry these donor template along with the CRISPR-Cas sequences, and then the donor templates will be cut out, and then this information will be copied uh, when the cells, yeah, they don't
What's
even.
amazing is that biology is amazing. You can put in this spaghetti, somehow with this system, it manages to pick out the right things and put them together appropriately because of the guide RNAs make a big difference.
Yes. And
So,
when you design the donor template, you also have a complementary sequence overlapping. So the cells know that these actually, are part of the cellular sequences. They are fooled. Sorry, John. Yeah.
yeah. Lynn has a question.
Yes, I do. I'm trying to understand the downside of this, and specifically, if you are not precise in how you cut, could you get unintended consequences like mutations or?
That's amazing question. I have slides on that actually. So
Oh,
I'll come to that and discuss in a few minutes.
thank
I
you.
just want to mention there are already 46 clinical trials going on using CRISPR for editing cells, especially immune cells. So I wrote a piece on conversation before on how you can edit white blood cells and especially immune cells, and then arm them to be now ready to fight against cancer. So there are many of these clinical trials are working on fighting cancer, and some of them are also working on diagnostics, as well as other disorders, genetic disorders. So blindness is one of the things. The genetic blindness is companies are working on that as well, because you can easily reach to the eye and inject CRISPR-Cas complexes. So I'll come to the problems with the CRISPR in a moment, but I just want to highlight where the technologies have built. I mean, there are so many technologies, and it might be very confusing to know all of them, but I want to mention that this was the preliminary technology of gene editing, where you can cut DNA and fix DNA. People have figured out ways to further improve that. So what they have done is they have shown that you can Cas is it's got two teeth. Okay, so you can break one of the teeth and it becomes like a single tooth Cas. Okay, and so you can nick DNA, so you don't have to make double-stranded cut. You can nick the DNA, and that has certain advantages also, because cells repair it differently when it's nicked versus when it's double-stranded cut. Other things people have done is they have made toothless Cas. So it's like the scissors are blunt, so it's going to bind and take your complex to the location of the DNA where you want, but it will not make double-stranded cuts. Okay, and so what people have done is they have used that type of information. It's called dead Cas or dCas9, and so the most common Cas people use is called Cas9, and the Cas9 is the one that was awarded Nobel Prize for Jennifer Doudna. And this Cas9 is the one. There are many different types of Cas in our body, but the Cas9 is a single effector protein, meaning it is only one protein. One protein is required instead of having many different proteins come together to make double-stranded cuts. And so people have done this dCas9 systems where the Cas, both the teeth or the nucleus domains are mutated. And so the dead Cas9, you can add any activator or repressor, and that can now repress any gene of interest or activate any gene of interest. So you can also add things like things to visualize where your sequences are. So for example, you can add a fluorescent protein, and so it will light up your cell here, and you can see where things are. You can also add genetic modifiers, so you can modify epigenetic modifiers, so you can modify the epigenome, which is a big field. People have also shown you can image, and also there are CRISPR-Cas systems that can cut RNA, not just DNA. So they were later discovered that there are Cas13 system, for example, which is analogous to Cas9, but it cuts the RNA. Then there is Cas12 system, which I'll talk about briefly, but Cas12 system also cuts DNA, but has a spatial activity. After it cuts the DNA, it becomes like a Pac-Man and starts cutting everything else. So people have, many scientists have been working, and they have figured out different types of Cas. And in fact, in my lab, we have 25 different new Cas systems that we have in our lab that have that are understudied or not even studied. So we are studying those. So it's this field is amazing. It's like the toolbox is increasing every day. Now the people have also figured out this NCas9 I was talking about, where it's one tooth. You can create a nick in the DNA, and what people have shown is that you just need to have the nick and don't need to do double-stranded cut, and you can add a base editor, meaning that the Cas will go to the site and then change single base letter from, let's say, from A to T, G, or C to T or A to G is how it changes usually. And so you can do you can fuse these with the base editors and just make single letter changes. People have also connected these with enzymes like reverse transcriptase, and they have shown that you can also generate in situ template. So Barb asked about how to deliver the templates. You can also in situ, I mean, you can, if you have a template within the CRISPR RNA, you can have a RNA, a longer RNA, and that RNA then gets reverse transcribed to DNA inside cells. And that technology is called prime editing, and it's been like the top, it was cited in 2019 as top 10 papers on, it's called prime editing, from David Liu's group, where I interviewed. So this, I mean, this also is from his group as well. Yeah, I mean, these are different ways, but the field is expanding, and I have not discussed here, like CRISPR can also do diagnostics, and that's a bigger, it's a very big field. And I would say it has helped a lot with COVID-19 as well in diagnosing, and my lab has been focusing also on that. So I'll talk about that briefly at the end. So I just want to show you, okay, let me skip the video for now, but I want to mention these are the problems with the CRISPR, and specificity is definitely a big problem. So these are 4 major problems I'm just discussing now, but there are many more. So first of all, the big problem is how to deliver these things inside cells. The cells, all these items are big, right? They don't want to go inside cells. So how to package them and deliver? And so AAVs have been used, as well as nanoparticle system and non-viral vectors have been used a lot, and people are developing them, and including my group. And so these are some of the papers from our group. And then CRISPR definitely has a problem of specificity. So if you want to target, the reason of non-specificity is because the bacteria has much smaller genome. So if a virus has even more, smaller genome, and when a bacteria is ready to cut the virus, it just has to scan the viral sequence, and it will make the cut. In case of human genome, we have much larger sequence of genome, and so in the genome. So the problem is that we have much higher chances of cutting it wrong. Okay, so we have so that is the sequence non-specificity. And in fact, if there are 5 mutations, it can still tolerate those 5 mutations, and it will make a cut. And so papers have been published where people have shown that you use one, it's a cutter for one site, and it may cut 100 different places, okay? And that's very bad. You don't want that. So my group actually, and others, many others, have been working on how to improve this. And the ways people have proposed is either you can modify the Cas so that it becomes a better Cas, and it does not tolerate, mutations, or you can modify the guide RNA so that it targets better. And so we found ways of improving that by shortening the length of this guide RNA, as well as by creating this hairpin loops. So we also made a light activatable version of CRISPR in the past. So the control is one big thing, because you can, people are also worried that, well, how would you make it specific to only one cell type and not cut other cells in the body? And so potentially light can be used, or many other triggers people have been working on, for example, ultrasound. Those triggers can also be applied here. So you can, the way we controlled using light was that we added these photocleavable, so we know the target region of the CRISPR RNA. We made a complement sequence to the target region, and this complement sequence, we added a photocleavable group. And so we added 3 of them. And so the idea is that when you shine light, they will photolize and you can turn on CRISPR. When you don't shine light, they remain blocked and they block the guide RNA. So you prevent CRISPR activity. So this is a turn on system. And we have also worked on turn off system, where you turn off the CRISPR, because people are also concerned about, well, what if the CRISPR is on always and it will make a lot of cuts? And the longer the CRISPR is turned on, the more chances of non-specificity you have, because we only have 2 copies of the DNA, target DNA in the genome. And then once the job is done, and let's say you injected 100 copies of CRISPR-Cas, once the job is done on those 2 copies of the DNA, you will see that a lot of other places it will start cutting, right? So we want to avoid that. So we have been also interested in how to turn it off, for example, using light as a way. And so other problem is the efficiency. So when you use CRISPR, not all the cells will be edited. And there's a problem of mosaicism as well in genetics, meaning that not all cells will be edited even if you do the embryo editing, okay? So how can you make sure that all the cells are edited is a big challenge. And there is a problem if you are using CRISPR technology for cancer therapeutics, for example. For example, what will happen is you can cut the DNA in the cancer cells, destroy the cells, and they will start shrinking. But then when the effect is over for the CRISPR, the cells, the cancer can relapse, right? So you can, and there has been studies focused on that as well. So there are problems on improving the efficiency, specificity, delivery, and control. Maybe not a big challenge, but yes, it can help. Yeah. Any questions on this?
I have a question. This is amazing stuff. There's, if you want control, let's say you're treating a cancer that is a consequence of stem or let's say blood cells generation in the marrow of the bone. Normally now you would get rid of all those cells and then introduce an appropriate cell to multiply. So and that could be, the question is the editing done in vitro or in vivo?
Yeah, that's a great question.
Yeah,
so most of the people, everyone is going through the easier route first, right? So they want to edit things first of all ex vivo. So ex vivo will be you take out the blood cells or the stem cells, and then do the editing and reinfuse back into patients, and that's going to be safer route because you're not injecting directly, and you can monitor how things are developing before you reinfuse. And so majority of the companies and also CRISPR technologies are focused on actually blood disorders. And so sickle cell, anemia, beta thalassemia, as well as blood-related cancers are the major ones people are focusing on. And then also solid tumors you can treat, but then they require you to train the immune system most likely, because it will be very hard to replace the tumor or shrink the tumor size down to 0. So
Right.
yeah.
Thank you. It's almost magic.
Yes.
Well, almost everything is magic. Yeah.
Julie.
Hey, John?
Yeah.
Hey, this is Julianne. I have John Axe here in the Oak Room, and he has a question.
Good.
Yes.
Thank you, Professor Jain, for a really interesting talk. I'm certain that this is very important, not only for medicine, but also improving food and livestock. And there's lots of money to be made in all this stuff as well. But I'm curious to know if I wanted to get into this, how much
Whoop. I lost.
We lost him.
We lost the sound. Maybe Julianne can repeat it for us. Yeah.
He started to say, how much would it cost to set up his own lab to do, what did you say, gene editing?
Yeah, is this an expensive thing, or can anybody, once they learn at that big center?
About $25, John.
Yeah, exactly. It's very cheap. So I will come to that about biohacking, because there are many biohackers who are really interested in using CRISPR for self-injecting and or like for treating their dogs, and people have done that already. And then there are groups who are high school students, right? And even middle school students now are starting to use CRISPR. I have actually advised a couple of students from high school who are doing amazing work using CRISPR. So again, the age limit will keep dropping. And because it does not cost a lot of money, because you can generate, I think it's around $150. You can get a vial of CRISPR-Cas complex. There's a company called Syntheco. You can order things through them.
The big worry in the biosecurity field is not what laboratories like Professor Jain's are doing. It's what somebody might do in their garage.
Yeah.
Especially with pathogens.
That was precisely my next question, because there are obvious beneficial things that you can do, but there are also malicious things that can be. And if people can have malicious intent, I was interested in how easily they could do that. So who's regulating this?
Yeah, it's very easy to do. I think if you have basic biology knowledge, just very, any grad student, for example, can do. High school students, if they know about biology, yeah, I know. It's all very simple, and that's why it's very scary as well. I'll come to that, and I'll talk about the ethics, and also I want to.
Okay, you can finish.
Yeah, I'm just wondering how much time I have, because I'm
about 5 minutes.
Okay, I'll take 5 minutes. Okay, so these are some of the companies and technologies people are working, some of the companies who are working on, and these are the 3 leading companies, CRISPR Therapeutics, Intellia, and Editas. And you can see what they have collaborated with big pharma companies, and they are working on sickle cell, beta thalassemia, immuno-oncology, as I mentioned. Then there is a transthyretin amyloidosis. I'm not 100% sure, but I believe it's a liver disorder. And then there is ocular for liver congenital amyloidosis, so for color blindness, and also then there are applications in sickle cell disease. So these are, you can see the market cap of these companies. So it's already multi-billion-dollar. And these are just 3 companies. There are 30 that are already there. So you can imagine, like, how, and all these are public companies, publicly listed. And so there is definitely a concern about all this, what's going to happen with this technology. So for example, Jian He, He is a Chinese scientist who actually used CRISPR to edit embryos for the first time. Right now he's in jail, but what happened was that he did not properly document everything. And there were a lot of, you must have heard this news, about this creating twin babies. So there are two, Lulu and Nana. They were born with the CRISPR editing. And um, of course, there has been, since then, there has been a concern on whether should we edit the CRISPR, should we edit the embryos with the CRISPR. He was a scientist in China, and he edited the embryos so that the babies become resistant to HIV. And so both the parents had HIV, and so it was almost certain that the baby will also be HIV positive when they are born. But then what he did was he said, like, certain groups of people have this mutation in a CCR5 gene, which makes them resistant to HIV, so that the HIV cannot enter. He caused that mutation using CRISPR in the embryos so that they become resistant to HIV. So again, like, there is a plus side of it, but there's negative side of using the using it in a baby, and it's ethically wrong because you are not asking the baby how they want to be changed, right? So
well, we don't know. I don't know what happened to those two girls who are
exactly.
now, if they're still alive, 4 or 5 years old. He went to prison.
Yes. So we don't know. And there was a news article, actually put a news, I have removed it, but there was a news article that they might have inadvertently improved their brain function also. So there was an article in Science, and then later in Technology Review, that discussed that, possibly if these babies are doing okay, they might have better brains. And so Vladimir Putin, for example, also had given speech, and that was also cited in Technology Review, that they can create, for example, soldiers using CRISPR with more muscular, more powerful, and also this concern about creating designer babies. Of course, where do we draw the line is the big question. Like, are we playing God? People have been editing also themselves, which I mentioned here, like as biohacking. So this is a very big discussion, I would say, ethical discussion, as well as whether you would use this CRISPR technology for editing patient. Is it okay? Whether you would use it for editing babies if they are going to be born with a disease without even asking them, and then whether you would be editing babies for creating uh, blue eyes babies or green eyes babies. Yeah. So yeah, that's definitely a concern. I would like to hear your opinion and thoughts on this.
Other questions?
I do see there is a
Julianne, are there questions on the chat?
no, but someone mentioned about Isaacson's new biography. I will talk.
Yeah, the code breaker.
Yes, the code breaker. So I'll show you also that. But these are some of the companies that are doing the gene editing in crops. So you can get better corn, wheat, tomato, sunflower, bananas, and soybean oil. Yeah, and in 2016 already someone launched a better corn. And then this code breaker. Yeah, so this is definitely a must read. I haven't completed the book, but Walter Isaacson, I actually met him in a conference with Jennifer Doudna. It was a gene editing conference. He appeared, and he was in a panel discussion talking about CRISPR. And yeah, so
I want to tell one personal story. About 20 years ago, I chaired a selection committee at the National Academy of Sciences to pick a young scientist under 35 for work in molecular biology. And of course, we selected Jennifer Doudna. So of course, there was a certain amount of personal vindication when she actually won the prize. And I wrote her a little note to that effect, and she sent back a nice little email.
Wow. That's amazing.
And next week, life gets more complicated for some of us, because next week we have the story of the black hole. And Jim Fry from the Department of Physics will give that talk. He's a very fine lecturer, but the topic is not so easy, I'm afraid. So see you all then.
Thank you, everyone. Thanks a lot.
And thank you, Piyush.
Thank
And I
you.
was delighted that you got here to the minute. That was very nice.
Thank you.
Thank you, everyone. Thank you for joining us, Doctor.
Thank you. Bye-bye.
Thank you very much.
Thank you. Have a great day. Bye.
Bye-bye.